Hello everyone, and welcome to our online lecture series for Bio1. Today we're going to discuss the circulatory system, an obviously very important system that has a lot of evolutionary variants for doing, ostensibly, a similar purpose. Just like with the respiratory system, we know that the circulatory system operates on diffusion. Again, the name of the game here is to get all of the nutrients that you need in, oxygen that you need in, as well as functions of protection and filtration going into the different tissues of the body while removing or carrying out wastes, carbon dioxide, which is produced as the byproduct of cellular respiration, and removing those things from your cells. Now, ultimately, if you don't need an entire organ system to do this, you are much more efficient. But there are limits to your complexity, to the amount of cellular respiration that you can perform. As with respiration, we do have those organisms that just rely on diffusion. Things like this jellyfish or cnidarians, as well as their cousins, the porifera or sponges. Now, without a true circulatory system, these organisms are fairly efficient and rely on having a low amount of calories relative to other organisms that do have circulatory systems, but they're of course limited in their complexity. Past these early animals of porifera and cnidarians, we see the evolution of two major different types of circulatory systems. The name of the game behind the nutrient exchange differs along whether your system is an open system, where blood does not stay in vessels, like those of insects like bees, and many other invertebrates, for those of you taking Bio 2, you'll be familiar with these as the protostomes, vast majority of them. And then the other variant that you'll be much more familiar with, because this encapsulates you, is our closed circulatory system, where blood does stay in vessels. And because it stays in vessels, it has a much longer distance to cover. And because it needs to cover that distance, that closed circulatory system necessitates a strong pump muscle. Or a heart. That heart is defined by having two different types of chambers. Chambers that receive the slow-flowing blood that is returning. Receive is one of those words that I will spell differently every single time. Not even sure if that's spelled correctly. The receiving chambers, or an atrium, and those that will propel the blood to move very quickly, the pumping chambers, called ventricles. Now, the ventricle that then forces the blood out of the heart is connected to very thick-walled vessels called arteries, because arteries carry blood away from the heart. The blood will then move with a high velocity through these arteries up until it reaches smaller and smaller carrying vessels. Now, arteries themselves are not doing a lot of nutrient exchange. They're just highways to deliver a lot of blood. The nutrient exchange is happening on the capillary level, which are small and thin enough for nutrient exchange. And last but not least, the blood will return to the heart at a much slower velocity in something called veins. And we'll end today's lecture by actually looking at the structure of each of these to see how arteries are able to deal with much higher velocity blood and how veins prevent flowback of slow-moving blood. The closed circulatory system also is characterized by the presence of a true lymphatic system. The lymphatic system is a way to deal with the non-blood cell liquids in the blood. Notably, because blood wants to live in a very pleasant environment filled with sugar, filled with warmth, all the resources you need to maintain a healthy body are flowing through that blood. As it turns out, it's really easy for bacteria and viruses to procreate in the blood, and that's a very scary prospect. The lymphatic system is there to catch and filter out and remove the viruses and bacteria that may find their way into the bloodstream. So closed circulatory systems will always have an associated lymphatic system. Closed circulatory systems also have something called blood pressure, which is incredibly important to monitor because you do not want too low blood pressure because then you're not delivering nutrients to your organs as necessary, and you don't want too high blood pressure because then you may be bursting the walls of your blood vessels. So blood pressure is very much a Goldilocks problem that's important to maintaining human health. Closed circulatory systems are present in most vertebrates or deuterostome animals. Those protostome and deuterostome will be vocab terms for Bio 2, not here, but I do like to make that connection. And last but not least, because I've gotten this question enough in other classes, we have a quick discussion on what a bruise is. A bruise is actually blood that has left the closed circulatory system. So there's been some sort of rupture in a artery, a vein more likely, not an artery. So veins can pop more frequently, and the blood will flow out to the surrounding muscle. Now, initially you will see that bruise as being blue-ish in color, blue to purple. Again, not because the blood is blue and low on oxygen, but rather because that is the light that's penetrating through the skin. Now, as that blood does begin to die off, it will then turn a brownish color, and as it's slowly but surely removed from the adjoining muscle tissues, it will turn yellowish before the bruise finally disappears. So the bruise is not evidence of us having an open circulatory system, as some folks have interpreted it, but rather a temporary rupture in our closed circulatory system. So let's explore a little bit more about the open circulatory system of insects and other invertebrates. Again, there is variation in this, but I want to introduce us to some of the major terms here. So open circulatory systems are found in invertebrates like bees and grasshoppers, and they are defined by having multiple hearts, without true chambers, but rather, I have to be able to read my handwriting, multiple hearts, without true entry chambers, but they do have holes in them called ostia. It is through those ostia that the blood that's present in the body cavity, which is literally just the hole that contains all of the other internal organs, the blood is floating around this. It is not separated from the liquids or the general liquids of the system. Therefore, it is not true blood, but rather lymph, a combination of the liquids that would be in the lymphatic system, as well as the heme-carrying molecules that would be present in red blood cells. And the hemolymph is actually mixing with interstitial fluids. All of the support fluids and other things that help keep the insect functional. So it's all kind of one giant soup. Now, because of this, insects cannot maintain as large of a size because of the amount of oxygen that they're getting to different tissues, and they in general have more devoted or less diverse functions of their circulatory system. Their circulatory system is defined by having some sort of dorsal blood vessel that goes along the back or the top side of the insect. The hearts are diffused along that dorsal blood vessel, and the ostia are facing in such a way so that way they can catch or funnel the blood from the entire body cavity and propel it along to different regions. The name of the game here is not to send blood in any particular direction, but to just keep it moving. By keeping it moving, you assure that diffusion will be able to run its course. It will at some point find its way next to those chitinous tubes attached to the spiracles through which the insect is breathing. It at some point will move adjacent to the digestive tract of the insect to pick up some nutrients, and it will also carry those nutrients and oxygen to the internal organs in the body cavity that need that. And in this way, the insect actually double dips in its movement. So movement of the bug contributes to circulation. Just like with the respiratory system, wherein movement helped, a movement of particularly wings or long-distance legs, helped move things so that way the spiracles could be filled with fresh air or higher oxygen air, and higher CO2 air is moved out. Movement in this case helps the liquid bounce around so that way it is more likely to have this balance, this greater balance of nutrients to wastes. Now, just like with the respiratory system, this open circulatory system is more energy efficient. So it's kind of important to address why insects would have to be more energy efficient, or why they would be because of the era in which they evolved. Today, we have, generally speaking, more calories available to us because of the primary production of plants. Fruits produce readily digestible calories. Fires help us cook down foods. The vegetative matter grows much quicker with much less defense compounds. I mean, think about a leaf of a pine tree versus a leaf of an apple tree. One is much tougher to digest down. And this reflects in the organisms that have learned to thrive during these eras. Insects date back to something like 415 million years at least. And during this era, there were no flowering plants. It would take another 300 million years for flowering plants to make their appearance on the planet. In this era of mosses, conifers, ferns, you would not have a lot of nutrients readily available to you. Therefore, these organisms would have been particularly energy efficient, thriving on fewer calories than their later evolving vertebrate counterparts. Now, of course, we still have our independent ecological niches that we sometimes have small amounts of competition for, but generally speaking, vertebrates and invertebrates are filling different ecological roles. And of course, as we discussed very early on in this class, we are potentially seeing a sixth major extinction event. And one of the canaries in the coal mine for this extinction event, beyond the drop in diversity of amphibians, is our drop in abundance of insects. There are just fewer bugs on the planet than there were 50 years ago, and that is causing a problem to our ecological systems. They do fill an important role, even if you think they're occasionally creepy and crawly. Insects do a lot of recycling for us, and some of their cousins, like spiders, help keep certain insect populations down. So it is important to study them and see what can be done to ameliorate the effects of anthropogenic climate change on insect populations. Because we did evolve in an environment that was filled with insects, performing that oftentimes role of recycling nutrients in our systems. But enough of that. They are more complex, and I do encourage those of you who want to go into insect biology or entomology to read further from your textbook. But now we get to discuss the evolution of the closed circulatory system. So for our closed circulatory systems, there's going to be a couple of drawing conventions here. So first I'll say that the next few drawings that we're going to go over are my simple cartoons of these circulatory systems, and then we'll have actual illustrations from the book as to how these circulatory systems are structured. So general convention here is that the arrow will be showing the direction of the blood, particularly the direction that it's being pushed through, and then the color is going to show whether it is oxygenated for red or oxygen rich, and blue for CO2 rich, even though we know that blood without high amounts of oxygen is not actually blue, and then purple for a mix of the two. So our first closed circulatory model is something that occurs in fish-like organisms. It's a two-chambered heart. Now, these two chambers also revolve around two major regions that they're going to be sending the blood to, either the gills or the system or the entire body. And now our heart is actually located not centrally, it is centrally to the fish, but not to the left side as we draw here. But we're going to, again, for the purposes of this cartoon, show this. The heart will always be some sort of box in this drawing, and it's got its two chambers. The receiving atrium, where blood came from the system to the atrium. And in fish, it looks much more like kind of just a small sac. Not really that muscular, not really that seemingly purposeful. It's just kind of a place for blood to pool. And then that blood will be pushed over to the ventricle. And you do have more muscle in the ventricle. From the ventricle, it will get pushed towards the gills. And then from the gills, it gets oxygenated. So part of the gills are deoxygenated as it enters, and then it is oxygen rich afterwards. Then the blood leaves the capillaries that are in the gills and makes its way to the system, where it is highly oxygenated up until it gets depleted, converts in the capillaries here of the system, of the entire body, to then make its way back to the atrium. This fish two-chambered heart has a one-loop circulatory system. that's defined by being unidirectional. It just goes in one consistent pattern. It's a little smaller so that I can fit everything on here. Our next model, as you'll learn in Bio 2, the next group of complex organisms, the first to arrive on land but still spend some of their life in the water, are our amphibians. And amphibians have a 3-chambered heart. In this case, still 1 ventricle, 2 atria, and more importantly, 2 routes. Or we start to see 2 kind of loops develop. Again, displaying the heart as a box, but in this case, a 3-chambered box. 2 atria, 1 ventricle, and the blood inside the ventricle, this time, is going to be a pool of blood that comes from an oxygenated, an oxygenated atrium and a deoxygenated atrium. This mixed blood will be pumped. So again, here our arrow for the pumping in the ventricle. It will be pumped to 2 different loops. One that goes towards the lungs and the skin to be oxygenated, and then return to the atrium. And then the other loop that will go towards the body, or the systematic loop, which will then return from the body, deoxygenated, to the heart. And because there are these 2 loops, or 2 routes, even though they kind of diverge in a weirdish way from 1 ventricle, those 2 loops are an important innovation, because then you're getting much more oxygen-rich blood being delivered to the body. And it is this 2-loop model that you inherit today, with some notable distinctions. So now we'll draw our mammal circulatory system. Again, an update in how many chambers there are. 4 chambers. 2 atria. 2 ventricles. 2 full distinct loops. Again, displaying the heart as a box. We have atrium, atrium, ventricle, ventricle. And importantly, now we can distinguish these. We have a right atrium and a right ventricle, a left atrium and a left ventricle. The right atrium and right ventricle are receiving deoxygenated blood. Therefore, they are pumping it to the lungs, which we call the pulmonary loop. The pulmonary circuit. This deoxygenated blood then gets oxygenated in the lungs and carries over to the left atrium and left ventricle. That left atrium and left ventricle then push this blood that's highly rich in oxygen towards the body, the systemic circuit or systemic loop, where it becomes deoxygenated and returns to the right atrium and right ventricle. This allows for no mixing of high oxygen and low oxygen blood, as we saw in the amphibian model, and therefore delivers the most oxygen to the mammal body. This system has a little bit of a difference in some of our early reptiles. You'll notice that the basic premise of a four-chambered heart actually evolved twice. Our reptile cousins do not have the same type of heart as us, but it is similar. They have a pseudo four-chambered heart. They have two atria and two kind of ventricles. Their ventricles have a gap in between them, meaning that there is some mixing of oxygenated and deoxygenated blood. Now, this 3.5 chambered heart seems to have led to the evolution of the bird's four-chambered heart, because birds are a subset of reptiles, and birds do have the same thing that we do with a truly four-chambered, completely separated heart. So, four-chambered heart seems to have evolved twice, both in mammal lineages and in the avians, the avians being a subset of the reptiles. Now, from my simple cartoons here, we can take a look at the illustrations from your book. The book shows a fish's circulatory system with a single ventricle, kind of looking like it's more a two-chambered thing, and a single atrium, and then flow to gills. In our amphibian heart, you have purple indicated here in the middle of the heart by the mixing of the two, and then it'll fall into one of two chambers, one that goes to the lungs and skin capillaries for oxygenation, and then another that goes to the body for oxygen delivery. In our reptiles, we have mostly a four-chambered heart, but we have that last bit of mixing that occurs, a septum that does not separate the heart entirely. And then, of course, we have a mammal's circulatory system with a truly four-chambered heart that does not overlap. The lines will, but the vessels don't in 3 dimensions. So keep in mind that that two-loop actually completely overlaps only in mammals and reptiles. In amphibians, you do have that one loop that will spread into 2 different delivering vessels. So hopefully either the professionally generated images or my cartoon drawings help make sense of these 2 different types, or of these different types of circulatory systems. The one-looped fish, the 2-looped amphibians, reptiles, and mammals, and then the 3-chambered amphibians, 2-chambered fish, and 4-chambered mammals and birds. I will say birds generally have a larger heart relative to body size because of their higher metabolism. Our next discussion in this lecture is going to be the actual constituents, the components of our circulatory system. Now, as you'd imagine, in terms of dry weight, the red blood cells are made up primarily of hemoglobin, and so hemoglobin is our biggest constituent of our circulatory system. We also have plasma in our circulatory system, which is the liquid in the blood, the non-cell component. It is a mixture of water, proteins, salts, lipids, looking at you cholesterols, and glucose. Lots and lots of sugar floating around in your blood. Your blood is also made up of cells. Both, red and white blood cells. More on those in a second. And then last but not least, platelets. These are cell fragments that help with clotting. And so these four major constituents, these three major constituents, your plasma, the cells, and the platelets, make up your blood. And the role of the blood, therefore, corresponds to those three, or if you count red and white as separate, different types of cells. First and foremost, the role of the blood is stabilize different aspects of chemistry, like pH, like osmotic potential, make sure that there's enough water, and temperature. So blood is there to make sure that all of your homeostatic factors are present. The pH that you expect, the temperature that you expect, and the amount of water that you expect. Number 2 is delivery of nutrients and hormones. Obviously very important to blood itself. The delivery system, blood itself, because blood is the delivery system of so many different things in our body. The third role for blood is to remove wastes. And then last but not least, fourth is to clot and protect. And so each of these different components of blood help achieve those goals. We're going to take a moment now to discuss a little bit more in detail because you may be familiar with those components from an earlier biology class. So we need to upgrade your understanding by looking at the different types of cells that go to make our different blood components of platelets, cells, red and white, and plasma. So the first one that I want to discuss are our basic origins of our red blood cells, our erythrocytes. Here we have a scanning electron micrograph image, so this is actually what they look like. Our erythrocytes, or red blood cells, are very interesting because, change the size of my pen here, they have no nucleus. They have no mitochondria. What? They also have no endomembrane system. No Golgi, no ER. They are really, really bare bones. Though I will say that it is mammal specific that they have no nucleus. And the reason why they lack all of these other internal structures is so that way they can be better deliverers of oxygen. They're just chock full of hemoglobin. That's most of what they're meant to be delivering. The balance here is between energy efficiency and oxygen delivery efficiency. You want to make sure that you have enough oxygen for your organism. Because mammals are so oxygen needy, we want to make sure that our red blood cells have the most space to carry the most oxygen available. In order to improve the diffusion of oxygen across the membrane, they have their surface area maximized. Because, they are biconcave. Or that double-sided disc that dips in the middle on both sides so that way they can absorb oxygen the most effectively. Now, they do have some proteins and other identifying features on their plasma membrane to identify to the rest of the body as to what they are. So they do have glycolipids and glycoproteins on the outside. For identification and communication. Now, the identification system for these erythrocytes is something that you're likely to be familiar with. The A, B, O blood groups, which are essentially different glycoproteins that have different sugar molecules sticking off the top of them to identify them as a very particular blood type. Last but not least, when discussing erythrocytes, we have to talk about their recycling, removal, and harvesting, essentially, because without a nucleus, you can only maintain for so long. So all erythrocytes are recycled in the liver. So erythrocytes are the vast majority of your blood. Again, they themselves being comprised of by 96% dry weight of hemoglobin. But beyond the erythrocytes, we do want to talk about the leukocytes. Which comprise only 1% of the blood, but are very important because they are our white blood cells. Now, leukocytes have a nucleus, defined by that nucleus. And in fact, their nucleus and surrounding features divide leukocytes into one of two groups, either the granulocytes, which have a grainy texture, or the agranulocytes. Now, you can see these differences, or the different types of cells that are granulocytes and agranulocytes, down here in this image from your textbook. Our granulocytes are our neutrophils, eosinophils, and basophils. And our agranulocytes are monocytes and our lymphocytes. Again, remember that cyte just refers to a specific type of cell, and each of these basophils, eosinophils, and neutrophils, well, they perform slightly different functions in the body, and those functions are beyond the scope here. I just want you guys to know what the different categories of leukocytes are, the granulocytes and the agranulocytes. Now, altogether, these leukocytes are present in the bloodstream to identify self versus not self, because they want to be dealing with our immune system, or they are our immune system. They want to identify things that should not be there and need to be killed. Our granulocytes are our first responders in our immune system. Their large nuclei allow them to process a lot of things, meaning that they're able to identify what should and should not be there, and then get rid of the things that should not be there. The granulocytes themselves are defined by having a grainy texture, which are actually secretory vesicles, meaning they have things at the ready to dump out to invaders that should not be there. Now, the contents of those granules are quite interesting. They can have antimicrobial agents, so proteins that happen to deal with microbes very well, some enzymes that happen to break down bacteria, specifically like the enzyme lysozyme, that's going to dissolve the cell walls of gram-positive bacteria. And then even some other things that you might be familiar with. Vesicles that have low pH, or acids to kill things, and then oxygen-derived products like hydrogen peroxide. So the same thing that we use on our cuts outside to kill bacteria present in a wound are present within our granulocytes. In contrast to being the first responders, our agranulocytes play a bigger role in adaptive immunity. So think antibody antigen, killing cells that are already infected, and a lot more complex response. You can also think of these as precursors to your major types of B and T cells. More on the immune system in a couple of lectures here. Immune system is very complicated and very cool. But that short description makes up all of our different types of leukocytes, which are our white blood cells. So with red blood cells, erythrocytes, and white blood cells, leukocytes, covered, we now can take a moment to talk about the plasma or serum. The important features we've already pretty much mentioned, the contents of it being mostly water, protein, salts, lipids, and glucose. The one thing I didn't mention is that it has molecules that buffer the pH of the blood. So I didn't want to leave that one out. And then, of course, we have our big cells, megakaryocytes, which go on to make our thrombocytes. Now, thrombocytes are made in order to be broken down. Kind of weird to think about, but the thrombocytes themselves go on to make cell fragments, or platelets. So they're absolutely ginormous, accumulate a lot of proteins, including proteins like fibrin and clotting or coagulation factors. And they need a whole bunch of vitamin K. So now I'll ask you to dig back to your notes on photosynthesis and tell me what vitamin K was from that lecture. Well, cue the Jeopardy music, or cut back in and have me remind you that vitamin K, or vitamin K1 at least, was phyloquinone. One of those proteins that was essential to the electron transport chain after photosystem P680, photosystem 2. So these thrombocytes are filled with vitamin K and fibrin and other clotting factors, and will go on to make your scabs. So you have the image from your book that kind of illustrates all the different blood cells floating next to one another with all the little platelets, the cell fragments made of fibrin, floating in between them. I did forget to mention that thrombocytes and platelets are mammal-specific. So you will notice that clots are made by a lot of different organisms, but the presence of these platelets floating through the blood are mammal-specific. Birds, for example, have monocytes that are floating around that will then begin the clotting process. So that actually has downstream effects to things like heart attacks, which is weird to think about. You can go ahead and read more about that if you like. But not all organisms have this readily available clotting factor that we find so useful in mammals. You have this image from a Wikipedia past, and then the more modern Wikipedia image illustrating all of the different cells, where they are, and what they come from. So we know that all of our blood comes from different groups of stem cells, myeloid progenitors or lymphoid progenitors. They then begin a commitment within, of course, this is all happening in the bone marrow because the bone marrow itself is actually making our different blood cell types. You then go from a totipotent or mostly potent stem cell to some sort of committed progenitor, like a megakaryoblast that will then progress into a pro-megakaryocyte that will then turn into a megakaryocyte, which that has to be one of my favorite technical biology drawings ever. Look how goofy and large that cell looks. That then breaks down into a billion pieces into our thrombocytes, our platelets. And this pattern of growth is consistent across all of our cells in that both white and red blood cells derive from the bone marrow, but then go to mature in different tissues. So hopefully, obviously you don't need to know all the intermediate steps. That's for an advanced medical class. Here is just an illustration so that way you can kind of keep track of things. So we'll now get to, I guess I forgot to mention what this is here. This being our segmented nucleus, so it's not that the cell has multiple nuclei, but that's a question that I get frequently. It looks like the neutrophils and the basophils have multiple nuclei, but they're actually just kind of connected very lightly in between them, still one giant nucleus, and this is actually a neutrophil. Kind of unique looking, so you can always tell apart your white blood cells by having nuclei and your red blood cells by not having nuclei. If only there were stains for nucleic acids that you might be familiar with from this class. All right, so let's get to the end of the lecture by discussing some basics about the heart pumping. So I don't expect y'all to be able to keep track of everything in an electrocardiogram or EKG or ECG. But I do expect you to know that a systole is the contraction of the heart, and that a diastole is the relaxation of the heart muscle. Again, being comprised of a cardiac muscle that's involuntary, the heart is able to beat for so many years without you ever having to think about it. The way an EKG reads is that first, an electrical impulse travels from the sinoatrial node to one of the walls of the atria, causing them to contract. So, squeezes blood from the atrium to the ventricle. Then you've got a little bit of prep work where the impulse reaches the AV node, which delays it by a fraction of a second, and that is very important, so that way it has a chance to fill. The filling occurs, and the bundle branches carry the signals to the AV node, to the heart apex, the bottom of the heart. And that bottom of the heart is where the contraction is going to happen. So you have a massive impulse where the signal spreads through the ventricle walls, causing them to contract. You need one giant pump to push both of your ventricles. And that's the basic readout of those, of the EKG from the medical shows that you'll oftentimes see. But keep in mind, I'm only asking you to keep track of systoles and diastoles. And then the very last thing is our illustration of our artery and vein. So from this grainy image, we see that the artery is on the left with multiple thick layers, the vein on the right. The inside of each of them is called the lumen, which may seem familiar from if you remember this term from the photosynthesis lecture. Inside of the lumen, you have a valve within veins that arteries lack, so veins have valves to prevent the backflow. The blood will be flowing this way, and then as it lowers in pressure and starts to slide back down, it'll actually close the flaps there to prevent the backflow. And then each of these are comprised of 3 tissue layers, with the tissue layers of the artery being much thicker, though they are present in veins. You have the tunica intima, or your intimate shirt. Tunic being shirt, intima being the one that contacts the blood. So that's an endothelial layer, a skinish layer on the inside of these blood vessels. You then have the tunica media, which is made of smooth muscles and elastic fibers to help things moving. And then last but not least, the outer protective layer, the tunica externa. so keeping all the blood vessel together. And hopefully with that, you have kind of a quick run through of what circulatory systems are built like, meaning where does blood flow, how are they different, and then some of the cells, the beautiful, beautiful cells that are, again, a major focus of this class, and give you a background, give you the necessary background in order to understand our future discussions of evolutionary biology.